everyone, it's Michelle, your CSEC Biology tutor. In this past paper solutions video, I'll be covering the CSEC Biology January 2019 paper 2, and I'll be looking at all 6 questions in this video. So let's get started with question 1. So part A, a sample of a meal was tested for the presence of reducing sugar and protein. Outline the procedure used to prepare the sample for carrying out the food test. So for solid foods, you're going to need to crush up the meal sample first and add some water, preferably distilled water, to it in order to form a solution before you can carry out any of the tests. And then you're going to add equal amounts of the food sample solution to clean, boiling or test tubes. So then this will prepare us for the next step. So to clean, since we have 2 different tests that we're going to be performing on the reducing sugar and then the other on protein, you're going to need 2 test tubes or 2 boiling tubes. Part B, complete table 1 by describing the procedure that is used to test for the presence of each food nutrient in the meal and the expected results if the food nutrient is present. So first of all, to test for reducing sugar, you're going to need to add the food sample to a clean boiling tube and add a few drops of Benedict solution. So Benedict solution contains copper sulfate, and you're going to heat it gently, heat this boiling tube with the solution. Gently, you're going to heat it in a water bath for a few minutes. So that heat is to generate the reaction. So if reducing sugar is present, you're going to expect to see a color change from blue to orange-red. And usually when it's orange-red color, there may often be a brick red precipitate as well. So that is for the reducing sugar. Now with the protein, so you're going to take another food sample, adding it into a clean test tube, and you're going to add a few drops of burette solution. So burette solution contains sodium hydroxide and copper sulfate. So burette solution would also be blue, similar to Benedict solution. In this case, there's no heat necessary. You're just going to shake the contents vigorously to mix the solution with the food sample. So after a while, if protein is present, you should have the expected color change from blue to purple or violet color. So that is the completion of that table. Okay, moving on to part C. Another meal rich in carbohydrates and protein was prepared and given to a sick child recovering from an illness. Part 1, name 1 enzyme involved in the chemical digestion of carbohydrates and 1 enzyme involved in the chemical digestion of proteins in the alimentary canal. So the enzyme which digests carbohydrates, a common one would be amylase, that is specific to starch. And then the enzyme for proteins would be pepsin. Part 2, suggest 1 reason why it is important for the recovering child to eat adequate amounts of carbohydrates, and 3 reasons why it is important for the recovering child to eat adequate amounts of proteins. So first of all, carbohydrates is necessary for the child to be able to release enough energy to help in the recovering process. So carbohydrates is known, this is a nutrient that is known to produce energy, which would be produced via respiration. So that is important. And then secondly here with the proteins, so the 3 reasons I have. So proteins are needed for growth and repair of tissue. So any worn out tissue, cells, proteins are necessary to help in the growth and repair of that. And then secondly, proteins help to build useful molecules in the body, such as hormones, which will regulate various activities, antibodies, which will be extremely important as is relating to the recovery process of the child. So assuming that the child is sick because of an infection, the antibodies will definitely be important. So antibodies are those protein molecules that help to fight off pathogens in the body. And then also enzymes. So all of these important molecules are proteins, and this will definitely be important in the recovery of the child. And then thirdly, proteins are actually also a source of energy. So it's usually not the first source of energy that the body would rely on, but proteins, in addition to fats as well, there are also other sources of energy besides carbohydrate. Part D. Green plants produce and store carbohydrates. Suggests two ways in which the type of nutrition carried out by green plants differs from that carried out by humans. So first of all, green plants carry out autotrophic nutrition, in which they utilize the energy from sunlight to make their own food. In humans, however, we demonstrate heterotrophic nutrition. So we cannot make our own food, and we must depend on other organisms in order to consume the nutrients that we need to survive. And then secondly, green plants will use simple or inorganic molecules such as water and carbon dioxide to produce simple organic food such as glucose. And green plants generally do not require mechanical digestion. Like us and humans now, we consume both inorganic and organic molecules. So the inorganic molecules would be like the minerals, and then the organic molecules would be carbohydrates, proteins, fats, vitamins. So these, and the inorganic molecules would also include water as well. So we consume both of these type of molecules as nutrients. And then in terms of the large nutrients, so going back to the carbohydrates, the proteins, the lipids, they will require both mechanical and chemical digestion, unlike in plants, which would only have the chemical digestion part, which involves enzymes. But as it relates to mechanical digestion, we as humans, we need to use our teeth to help break down the food into smaller pieces before it can be broken down by enzymes. Part 2, describe the procedure to test for the presence of starch in a leaf taken from a green plant. So first of all, you're going to take the leaf from the plant, place in a beaker of boiling water, and this will be on top of a tripod with a Bunsen burner. And this step is to destroy the cells of the leaf. Secondly, remove the heated leaf and place in a boiling tube of ethanol, and that boiling tube will be placed back into the warm water bath. And this step is necessary to extract the green pigment chlorophyll from the leaf. Thirdly, remove the leaf from the boiling tube and place on a white tile or a petri dish. And then you're going to carry out the last step, which is adding a few drops of iodine to the leaf, and observe the color change to blue-black if starch is present. Part 3, explain why plants store most carbohydrates in the form of starch instead of in the form of a reducing sugar. So starch is the energy storage molecule in plants. It is a complex carbohydrate. It forms in soluble grains, so it's not going to be dissolving in water. So these insoluble grains in the plant cells are necessary for storage, but then it can also be broken down and converted into glucose, which is the reducing sugar, and that happens when the plant needs it for energy. Now, the problem with reducing sugar, such as glucose, is not good for storage because it's soluble and it's highly reactive, meaning that it can easily take part in chemical reactions within the cells. So glucose will not last very long inside the cells as a result. So starch is a better storage molecule inside of the plants rather than reducing sugars. Part 4 suggests 2 reasons why it is important for plants to store starch. So first of all, in the event that the plant is exposed to harsh conditions such as a drought, and it cannot obtain the necessary raw materials, and we're talking like water specifically, and the plant is unable to carry out photosynthesis, it will still have a food storage there to rely on for energy. And then secondly, plants also store starch in these vegetative organs such as root tubers, you have rhizomes, runners. All these organs can provide energy for asexual reproduction, for the producing of new offspring plants. So that is what is known as vegetative propagation. So those are two reasons why it's important for plants to store starch. And not all plants will be able to carry out asexual reproduction, but that is as it relates to those type of plants. So that is the end of question 1. Okay, let's move on to question 2. Part A1, name 2 biotic factors and 1 abiotic factor that may account for the extinction of a species. So biotic factors are the living components in the ecosystem. So plants and animals, they will provide food sources for organisms. And then the abiotic factor, a very important one, will be water, which is necessary for survival of all living organisms. Part 2, state 2 activities of humans which may have a negative impact on species diversity and 2 measures that may be taken to reduce the impact of these activities. So the first activity I have is deforestation, the cutting down of trees, reducing the number of plant species in addition to animal species. So removing trees destroy the habitats of various animals. Secondly, pollution in general, but I'm focusing here on air pollution. So the release of the toxic gases from factories, vehicles, these can harm the respiratory systems of animals and affect gaseous exchange in plant species, so leading to the death of these organisms. So measures that can be useful in reducing these outcomes, reducing the impact of these activities. Afforestation is important, planting more trees and reducing the cutting down of trees for clearing of land or for building materials. And then secondly, using alternative energy sources such as solar energy, wind power, biogas. These will help to provide power for our industrial processes, and also using non-toxic lead-free fuels in vehicles rather than burning fossil fuels, which can be very damaging to the environment. Okay, let's look at part B. The graph in figure 1 shows the growth of 2 populations of spider mites, one which feeds on plants and one which is carnivorous and preys on the plant-eating spider mites. So we see on the y-axis the number of organisms, on the x-axis we have time. So basically we're seeing this fluctuation of peaks and troughs for both populations. So it says to label on the graph the curve which represents the predator using the letter A. So the predator population would be the dotted line, the dotted curve. So always remember that the predator population would generally overall be lower than the prey population. So therefore the solid line would represent the prey population. So this all relates to the transfer of energy in the ecosystem, and usually the prey population would be the ones that would have more energy in the ecosystem than the predator. So generally the prey population would tend to have a larger numbers compared to the predator population. So that is how you can identify that the dotted line will be the predator population, while the solid line will be the prey population, which have higher peaks. So secondly, part 2, use the number of organisms and the shape of the curves to state 2 reasons for your answer in B1. So as you can see from the curves, the predator and prey populations have an influence on the numbers of each population. So each population influences the other. And the graph is showing a fluctuation in predator and prey population numbers with a similar pattern of peaks, as you can see the top parts, and then the troughs, which would be the dips. So you can clearly see the similar pattern, and the two reasons for the answer. So first of all, the prey population increases due to favorable conditions. So when they have a lot of food available for them, water, the environment is ideal for them, and you will see that there's this increase of the prey population while there are few predators. Now secondly, the prey population would tend to reach a peak level, so this is the top of the curve, and then it decreases as the predator population starts to increase, since they now have more food available. So you can see that here in the diagram. So we have the peak level of the prey population when the level of the predator population is still low, but then beyond that peak you're noticing that the numbers of the predator population are actually rising. So it reaches that peak, the prey population reaches a peak, and then it starts to decrease as the numbers of predator start to increase. So you're seeing this similar pattern throughout the graph. So part 3, assuming that the population of the predator drops to 0, extend the curve of the prey population on the graph. Assume that no other changes have occurred in this community. So if the predator population is 0, though that is ideal for the prey population, so there's no need to worry about being eaten up. So you would expect that the prey population would actually have an exponential increase. There's nothing really there limiting it. So the prey population would increase, but then you can imagine that after a certain point of increasing, that it may eventually peak again, because when you have a lot of organisms in a population, that increases competition for various things, space, nutrients, whatever the case may be that they need for survival. So you could consider the fact that yes, they would have that exponential increase because the predator population is very low, 0, so there's no predators to worry about. But after a while, this overpopulation can lead to increased competition, and then eventually they would have a decreased number of preys in the prey species in the population. So for here, I'm just going to leave it at this level. So you're seeing that in that continuous increase when the predator population is 0. Now the next part of the question says, assuming that the population of the prey drops to 0, extend the curve of the predator population on the graph. So once again, assume that no other changes have occurred in this community. So if we have the prey population dropping to 0, that would affect the predator population significantly, because the predators depend on the prey. So if there are no prey in the population, there's no prey in the community, then what are the predators going to feed on? How are they going to survive? So 0 prey would lead to a decline in the numbers of the predator population. So that is what's shown there by the dotted line. Alright, so that completes that part of the question. Okay, let's move on to part C. A farmer planted 50 seeds of corn on a plot of land and 100 seeds from the same batch on a neighboring plot of the same size, hoping to reap twice the yield of corn. He gave both plots the same amount of fertilizer and water. However, the yield of corn obtained from the plot with 100 seeds was much less than that of the plot with only 50 seeds. So suggest 3 possible reasons for the lower yield obtained in the plot with a larger number of seeds. So the first reason there, since there are more seeds planted in the same plot size, so that's going to be less space now more seeds, same plot size, but it's going to be less space between the seeds. So there's less space available, and the seeds are not going to be able to germinate and develop a proper root system with such limited soil space. So that is one reason for the lower yield. Secondly, because also of the limited space, so now we have all these seeds, and if they do start to germinate, they're going to have a lot of increased competition for essential nutrients, water in the soil. So that is a problem there with competition. So we have competition for space, not enough space, and now we're having competition for nutrients. And then thirdly, more seeds, once again, due to the limited space, that can also lead to decreased oxygen availability. So it's less likely for the seeds to grow into seedlings with limited availability of oxygen, because oxygen is necessary for respiration, so that can reduce their growth and development. So these are three reasons why having more seeds in the same size plot would lead to lower yield. So that brings us to the end of question 2. So question 3, part A1, what is the effect on the expression of phenotype for continuous and discontinuous variation? So for continuous variation, you're going to have a wide range of the particular phenotype where there are extremes, and then there are the intermediate traits. So key thing to note with continuous is a wide range of the particular phenotype. So you have one extreme or the next, and then in the middle you have those intermediate or average expressions of the phenotype. Discontinuous variation now is different in the sense that you have distinct categories. There are no extremes and there are no intermediate traits. So you either fall into one category or another. Part 2, state how the data for continuous variation should be presented graphically. So for continuous variation, you can use a line graph or curve, or you may also use a histogram. So the histogram has the bars touching. Part B, state one characteristic of human beings that shows 1. Continuous variation. So height is a common example of that. So in a population, there are individuals with wide range of heights. So you go from the shortest individual to the tallest, and then you have the intermediate heights, the average heights. And then with discontinuous variation, blood groups is a common example. So you have 4 specific blood groups. You have AB, A, B, and O. So individuals in the population would belong to one of these 4 blood groups. So they are distinct categories. Part C, state 2 cellular components of blood. So we have the red blood cells and the white blood cells. So red blood cells for transporting oxygen, white blood cells are part of the immune system, and they help to fight off infections. Okay, part D, a couple with blood groups A and B has a son with blood group O. Complete the genetic diagram below to show how this is possible. So the parents' phenotypes are blood group A and blood group B. Now in order for these parents to produce each son with blood group O, each parent has to have the O allele present in their genotype. So we know that there are 2 possible genotypes for someone with type A blood type, but it has to be AO and not AA. So each parent has to have the O allele. So this parent would be AO for blood group A, and this parent would be BO for blood group B. So we have the gametes A and O, and then B and O. Now to carry out the cross, so we have the gametes up here for AO, and then on the side for BO. So you're just going to perform the normal cross using the Punnett square. So AB is the first blood type, the first genotype for the first child. The second child has BO. The third child would be AO. And the fourth child would be OO. So basically we have blood type AB. So in terms of the phenotypes now, I just went through the genotypes, which are the actual alleles, the genes that the children would have. But AB would produce blood type AB, BO would produce blood type B, AO would produce blood type A, and OO would produce blood type O. So those are 4 different phenotypes produced by this cross. Part 2, what is the genotype of the child with blood group O? So we just worked it out. So this is the child with the blood group O. He has 2 O alleles. So in order to be type O, in order to have that blood type O, you have to inherit both O alleles, an O allele from each parent, the father and the mother. Okay, part E. 2 sisters ages 15 and 16 have the same biological parents and were brought up under the same conditions in the same home. The 15-year-old is much taller than the 16-year-old, suggests 3 events which take place during sexual reproduction, which may have led to the variation in the height of the 2 sisters. So we're looking at 3 causes of genetic variation, which is produced by sexual reproduction, produced in sexual reproduction. So first of all, we're going to look at the crossing over of homologous chromosomes from the mother and the father during meiosis. So this is going to lead to an exchange of genes that form recombinant chromosomes. So the genes for height can therefore be different with each sister depending on which genes they would inherit. Secondly, random segregation or independent assortment of the chromosomes. So when they're separating and moving away to opposite ends of a cell, you can have some differences being brought about there. And then thirdly, fertilization. So the sperm from the father and the egg from the mother will carry different genes. So during the fertilization, a new combination of genetic information is formed. So the height for each sister would be determined by the type of genes found in the sperm and egg that produce them. So that is the end of question 3. Let's move on to question 4. Okay, so we're on to section B now. So question 4A, part 1. With the aid of a label diagram, describe how the mammalian heart pumps deoxygenated blood from the cells in the body to the lungs to become oxygenated. So I have the diagram here. So deoxygenated blood is coming from the cells, the various organs of the body, and this blood is going to enter the right side of the heart. So we have the right side here, and it's going to enter the superior vena cava coming from the upper body. So that is the largest vein in the body, the vena cava, and we have two types, the superior vena cava from the upper body and then the inferior vena cava from the lower body. So both of them are carrying deoxygenated blood towards the right side of the heart. So that blood is going to enter the right atrium. When the right atrium contracts, it's going to cause the blood to pass through the tricuspid valve. So the tricuspid valve separates the right atrium from the right ventricle, and then that blood is going to be pumped from the right ventricle when the ventricle contracts up into the pulmonary artery. So this pulmonary artery is going to connect to the lungs, and this is where the blood now would pick up the oxygen that is needed. So when that deoxygenated blood reaches the lungs, it collects the oxygen, and then it now can return to the left side of the heart through the pulmonary veins. So this is just the written description of what I just explained. Part 2. The structure of arteries and veins differ from each other. Explain how two of these differences allow these blood vessels to carry out their functions. Now, arteries have a narrow lumen, so very small lumen, and they have thick muscular walls to accommodate high-pressured flowing blood, which is coming from the heart and needs to be delivered to all the organs and tissues of the body. So those thick muscular walls help to withstand that high-pressured flowing blood. And then the veins will have a larger lumen, and usually the walls will be much thinner because they don't have to withstand high pressure. So we have lower pressured blood flowing through the veins from the body's organs towards the heart, and then we need valves in these veins to prevent backflow, because the blood is flowing at such low pressure, is more likely for blood to flow backwards. So therefore we have to have these valves present in the veins to stop that backflow. Okay, part B. Green plants also have specialized vessels such as xylem to transport water to their various tissues and organs. Part 1. State one similarity and one difference between the structure of a blood capillary and the structure of a xylem vessel found in the stem of a green plant. So the similarity, both the blood capillary and the xylem, they're both small, long, narrow tubes, and they carry fluids to transport materials. So we know blood is running through the capillary, and then we have water with dissolved minerals going through the xylem. So they're both carrying fluids to transport materials, and they are made of small, long, narrow tubes. Now the difference, blood capillaries are made of living cells. They consist of living tissue, while xylem is composed of dead cells. So the importance of the dead cells of the xylem is to prevent the water that is trying to transport from stopping and reacting with the cells, because living cells require water to carry out different chemical reactions. So that's the reason why the cells of the xylem are dead. Okay, part 2 suggests two reasons why green plants do not need to depend on xylem for transporting oxygen to their cells. So first of all, green plants obtain oxygen from the atmosphere through the stomata, so similar to carbon dioxide. So both gases can enter and leave through the stomata in the leaves, and the oxygen can then be transported to different cells in the plant through simple diffusion. So there's no need for special transport vessels to carry oxygen to various cells. And plants generally, they don't need these vessels because they don't have as high a demand as in animals, which are more active. So they're not going to need oxygen as quickly and as in such a high demand as it is with animals. So there's no need for special transport vessels in the plant to carry and transport oxygen. So the xylem, remember, is only responsible for transporting water and minerals, not oxygen. So oxygen simply gets in through the stomata and it can diffuse throughout the different cells of the plant. Okay, let's look at question 5. Okay, part A. Diseases may be categorized as pathogenic, represented by P, when they are caused by a pathogen, and hereditary, represented by H, when they are caused by faulty genes. Classify the following diseases by inserting either P for pathogenic or H for hereditary in each bracket. So we have yellow fever, which is a pathogenic disease. So is measles. And then sickle cell anemia is a hereditary disease, and so is hemophilia. Part B. John and his friend were bitten by Aedes aegypti mosquitoes while visiting an area in which many individuals had tested positive for dengue fever. John did not contract the disease, but his friend did. Part 1. Describe how the mode of transmission of dengue fever differs from that of the human immunodeficiency virus, HIV. Use this difference and your knowledge of the immune system to suggest 2 reasons why John did not contract dengue fever. So first of all, dengue fever is transmitted through the bite of the Aedes aegypti mosquito. So the mosquito has to bite you. The mosquito that is carrying the dengue virus has to bite you in order for that virus to get into the bloodstream, and then the virus would multiply, and then that's what leads to the symptoms of the disease. Now with HIV, it is different. The transmission is different, and they have multiple modes of transmission. HIV can be transmitted through sexual contact, which is the exchange of semen and vaginal fluids, through mother-to-child transmission, so through birth, although those chances are reduced in recent times. And then there's intravenous drug use, the sharing of needles, along with blood transfusion with infected blood. So there are multiple modes of transmission for HIV, but you cannot get HIV through the bite of a mosquito. So that is one thing that you need to be clear of. The mosquito cannot carry the HIV virus. It's only carried in human fluids, blood, vaginal fluids, semen, those type of fluids. So HIV attacks the lymphocytes when it gets into the blood, and this causes the breakdown of the immune system, reduces the individual's ability to fight off infections. Now, in terms of the reasons why John did not contract dengue fever, 2 reasons. First of all, the mosquito that bit John may not have even carried the dengue virus. So yes, the Aedes aegypti mosquitoes are known to carry the virus, but not every single one of them may have the virus within them. And then secondly, as it relates to John's immune system, so he may have a stronger immune system, which is better at resisting infection and fighting off the virus, or perhaps John might have had dengue already, and his immune system would have recognized the same strain of dengue virus that he would have had before. So therefore, even if it gets into his system, the immune system would recognize it quickly and cause the immune response, and therefore John would not experience the actual symptoms of the dengue fever. So those are 2 possible reasons why John may not have contracted the dengue fever. Part 2. Compare the treatment method normally used for viral disease with the treatment method normally used for diabetes, and compare one preventative measure normally used for viral disease with one preventative measure normally used for diabetes. So viral disease is an infectious disease. Diabetes is a chronic, non-communicable, so non-infectious disease. So with viral diseases, the best form of treatment would involve using antiviral medications to actually destroy the virus. Now with diabetes, insulin injections are usually administered. Other forms of treatment can also include medication as well to reduce the blood glucose concentration, and then eating a healthy diet. So those are alternative forms of treatment for diabetes. And then in terms of the preventative methods, so for viral disease, to prevent a viral disease, it's best to avoid places where, so for instance in this case with the mosquitoes, avoid the areas where the vectors of the disease are prevalent to reduce the risk of infection. And then for diabetes, reducing sugar intake is important. Also exercising, getting an active lifestyle. But reducing sugar intake is probably one of the key ways to prevent diabetes, because diabetes is as a result of high blood glucose concentration. Okay, part C, outline 2 methods used to control the mosquito vector, and for each method state the stage in the life cycle that is targeted. So the first method simply would be getting rid of breeding sites. So this would include tires and containers lying around that are collecting stagnant water. So mosquitoes like to lay eggs on the stagnant water. So this is going to target the egg stage, where it will prevent the adult mosquitoes, the female adult mosquitoes, from laying eggs on the water. So if you get rid of those breeding sites, you cannot have eggs being laid. Then secondly, fogging, or the use of insecticides to actually destroy the adult mosquitoes. So that is the stage targeted, the actual adult mosquito stage. So therefore, if you destroy the mosquitoes, that can reduce the breeding as well. So that is the end of question 5. Okay, question 6, part A. Outline one major function of each of the following parts of the human brain. So we have the cerebrum. So the cerebrum controls many functions, including memory, thought, logic, reasoning, motor activities, sensory functions. So I just chose to write memory. The cerebellum controls balance and positioning, also muscular activity. And then the medulla, or the full word medulla oblongata, that controls involuntary actions such as breathing, heart rate, and peristalsis. Part B. A motorcyclist crashed into a wall on his way home from a party at which he drank 5 beers. After determining that he was alive, the paramedic at the scene of the collision checked the motorcyclist's pupil reflex. Complete the flow diagram in the following box to show the sequence of events involved in the pupil reflex. So we have this pathway here. So the light stimulus, the light enters the eye, so it's going to enter the pupil, and it's going to go to the back of the eye where the retina is. So you have the receptor cells in the retina, and then the information will be passed on. So it's converted from light into the nerve impulses. So those nerve impulses, electrical impulses, are going to be transmitted through the sensory neurons. Then the sensory neurons are going to pass on the information to the relay neurons in the brain, and then that information will be passed to the motor neurons in the optic nerve, which connects the brain to the eye. And then in the iris, which is made up of muscular tissue, the circular muscles would contract, so that causes the pupil to constrict or get smaller. Now in the case if you place radial muscles here, if the radial muscles contract, then the pupil would dilate. So either one could work. Part C. Suggest 3 conclusions that the paramedic may make if the accident victim is alive but shows no response to the pupil reflex test. So using the pupil reflex test, the paramedic will shine the light to see if the pupils constrict and dilate. Now, first of all, the paramedic may think that the victim is unconscious, is passed out. Secondly, he may think that the state of unconsciousness may be due to some brain injury, optic nerve damage, so something related to the brain. And then thirdly, the paramedic may think that the state of unconsciousness may have been caused by high levels of alcohol in the victim's blood. So he might have smelled alcohol on the victim's breath, perhaps, and could have concluded that the victim was intoxicated. Part D. Explain 3 physiological reasons why the consumption of excess alcohol could have caused the motorcyclist to crash. So alcohol consumption for sure will affect the brain functioning and can cause the following things to happen. So first of all, he may have had slow reaction time, so reduced reflexes, muscular response. So this could have caused him to be too slow to react quick enough to press the brakes in order to stop fast enough. So a slow reaction time. Secondly, blurred vision. That's another effect. So this would have impaired his ability to see clearly while he was driving. And then thirdly, sleepiness or drowsiness, that could have caused the driver to even fallen asleep at the steering wheel. And then some other reasons: poor concentration, loss of coordination, handling the steering wheel. So any of those could be possible physiological reasons why the motorcyclist would have crashed. So that brings us to the end of this paper.